In this module, we will talk about the organization of a fermentation process. Before talking about the organization of a fermentation process, you know that in previous modules we discussed about different types of fermentation. So regardless of the type of fermentation, an established process may be divided into different basic components during the fermentation process. So there are, I have quoted here, the 7 different component parts in a fermentation process. Number 1, and very important, and you know that the mass culturing of the organism, I named the organism as the hero of a fermentation process. So without the hero, nothing is there. And then is a fermenter, which you see that stage on which the microorganism act or grow. And the third, very important, that is the media. So formulation of the media is very important component part of the fermentation process. And very critically, and fourth part, that is sterilization. Sterilization is very important because when there is a pure culture, then we can hope for our good production. But when there is a contamination, we cannot have a good production, because when there is a contamination, then contaminants can share the nutrients during the fermentation process. So the sterilization is very critical component part of the fermentation process. And then is a monitoring and control. How you monitor the process? During the fermentation process, we have to keep an eye on the fermentation process, that our product is forming, what are the pH, what is the temperature. So we have to run the fermentation process in a very controlled conditions. So the 6th step is recovery of the product. Because if you produce something, then there is a task to recover that product. So the recovery of the product is very important, and that is also the base of the fermentation economics. So if you produce any product and you cannot purify that and you cannot recover that, and then you have to be very careful about your product. And then last, when you purify any product and you extract some product from your fermented broth and remaining your effluent, so you cannot throw into the environment without the treatment. So you have to be very careful about the effluent treatment, because during the fermentation process, we have to keep in mind the containment level, because during the fermentation process, different type of microorganisms can be utilized. Either they are environmental friendly or they can be pathogens. So in case of pathogens, we have to look about the containment level. So in this picture, you can see that different steps involved in fermentation process. Start from on the left side from the stock culture, and then is on a small scale in which you grow the medium, and then there is an inoculum development, and then there is a seed fermenter, and then that goes into the production fermenter. But below there is a medium, raw material, and then formulation, and then sterilization, and then you put in a seed fermenter as well as in a production vessel. So next is when there is a specific time period, and then you harvest in the form of culture fluid or fermented broth, then you separate. So to separate the biomass from the other fermented broth, and then you harvest your product, and then gradually you purify your product, and then the packaging and marketing, and then there is effluent treatment. So this major process of fermentation, in which the propagation of the microorganism, and then the major which we call as the fermentation process or a production process, and the third major is the separation and purification method. So this is the whole organization of a fermentation process, apart from the types of the fermentation. As we see in the whole fermentation process and the organizational steps of the fermentation process, the whole and all the component part of the fermentation process can be subdivided into 2 sub-processes. One is upsteaming and other one is downsteaming. Upstreaming involves the steps from 1 to 5, and then downstreaming involves the steps and the component part from 6 to 7. So now in next slide we will see that what is upstreaming process. So actually the upstreaming process started from the inoculum development and all those parts and the steps involved up to the production of the product. So in which the stock culture, inoculum development, media development, inoculation, sterilization, and then product formation, they all come in the upstreaming process. So when there is a product and then you complete your fermentation process, then the downstreaming process starts. So the downstreaming process actually is the recovery of the product and then the treatment of the effluent. In downstreaming process also involve different steps in which first of all there is a separation from the solid to the liquid or liquid from the solids, depend upon where our product. And then we use different chemical and physical methods to purify our product using chromatography techniques, filtration techniques, either change in pH, either precipitation techniques. At the end when we purify our product, and then there is a special, you see, techniques by preserving those products with respect to their shelf life. And then after preserving them and after doing all the purification steps, then there is keep in mind the shelf life. We have to supply those fermented products where there is needed. So that is the transportation. But as concerned the downstreaming, that only involved up to the purification. The further steps are not involved in this downstreaming. So but the major step along with the purification, that is the effluent treatment. So that can be done by different chemical, physical and biological methods, because that is very necessary part of the fermentation process. So after upstreaming and downstreaming process, then there is a whole organization of the fermentation process is completed.